want a dye that can work with minimal processing. So I want a dye that anybody can use, that my grandmother can use, right?
Dr. Mireille Kamariza focuses on detection of tuberculosis. To tackle such a large issue requires determination, resilience, and perspective. Luckily, she's an expert in all three. In this Season 2 episode of Bringing Chemistry to Life, we speak with another member of the Chemical and Engineering News' 2020 Talented 12 about their work and trends in their field. I'm your host, Dr. Paolo Braiuca from Thermo Fisher Scientific. We began by asking Dr. Kamariza about her homeland and how she found her career in science.
So I was born in a small country called Burundi in East Africa. It's a landlocked country right at the heart of Africa. It's a war-torn country. And so when I was growing up, very early, in my life, the idea of doing science was far from my mind. I was, however, curious. I was particularly curious about the sky and what was behind the sky. And, as with any 4 or 5-year-old, you go outside, you play, you see animals, and then you look up and you see stars or something if it's at night. And I used to wonder a lot about what's on those stars. Like, can I walk there? Can I get there? Can I fly there? Right? But that was about the extent of it. My life was surrounded by chaos, right? Like the country was... In the early 90s, my country went through a huge turmoil, and we had to flee my country, and we had to move to a different country, and so like all the issues going with being a refugee in another country and just sort of assimilating in another culture, and eventually I was fortunate that my family, my siblings rather, and we ended up moving to the United States. It's fair to say that my life before and my life after drastically changed. When I moved here, I still didn't really think about being a scientist. It's not a dream that is part of the landscape for African immigrants who are in the U.S., right? There aren't many role models. But I had a really curious mind, and I had an inclination towards the sciences. And so when I was privileged enough to be in school at a community college, I took a lot of science courses, and I'm very fortunate that one of my early chemistry teachers took a liking to me and believed in my potential. And so she's the one who essentially said, I think you're a really good scientist. I think you should do research. I was like, what is research? I don't have time to do that. Do they pay? And it really was by way of her mentorship and guidance that I found my way through the door of scientific enterprise.
With someone with such a unique story like yourself, I think these are important things to kind of discuss about. At least for me, I'm really interested in that. So I just want to go back to the fact that we were thinking about the serendipity of your development and what happened to you, and meeting the right people and the mentors you've had in your path has been quite key actually to even make you realize that you could be a scientist, right? So when was the time when you actually started thinking about planning your scientific career? So when did you realize, I could actually do this? This is what I can be?
It's a very good question. I started at community college, as you said at the beginning, and my mind in community college was, let me just get a degree, get a job that pays more than minimum wage. And when I was taking the science courses, I met with a professor who sort of convinced me that the best way to get a good paying job is to transfer to a foreign university and get a bachelor's degree from a reputable college. And I was living in San Diego, so the school around me was UC San Diego. So I transferred to UCSD, and as part of that transferring process, she said, there are opportunities to do research and you can get paid for this. and I think you have the mind to do science, and this is a great opportunity that you should go for. And I did that, and it was an outstanding scholarship program for undergraduates at UC San Diego that I took advantage of. Somewhere along the way, I started liking what I was doing. I actually really enjoyed the science I was doing, and I nearing the end of my undergraduate time, I started contemplating whether just leaving science to get a job somewhere was really what I was attracted to. I had found my passion now in science. I knew a lot about molecular biology, and I just wanted to do more of it. I wanted to stay in academia, and I wanted to continue exploring this curiosity that I had about certain things in molecular biology. I still didn't think that I was a scientist, though. I just thought that I liked what I was doing. I didn't know I could go to grad school. Back then, the universities would have all these like requirements to apply and be admitted to a PhD program. And I just felt that I didn't fit any of them. And I would look at past PhD students in various programs, and none of them had a background like mine. So I really didn't think that this was something that I could do. My mentor at UC San Diego, Tracy Johnson, she is a Black woman, and she understood to a deeper level what I was going through. And she used to tell me, and she's like, none of them look like you, but it doesn't mean that you don't belong, right? That you're not going to be just as good as them if you get there. And plus, what does it take for you to apply, honestly? Like, what's going to take out of you? Maybe a couple of weeks putting together materials, and then you can go on your merry way if it doesn't work out. You should always shoot your shot, because if you don't shoot your shot, then you'll never know if you could ever gotten it. As you know, my little shy kid, I like timidly put together stuff. I was like, I don't believe any of this, but I did it anyway. And I got into the same school that she went to grad school. And I think that's what solidified it in me. The fact that she had gone through this first. She had come out the other end with a PhD. So that bodes well for me. And so I ended up choosing Berkeley because of that. Because that it was a place that she spoke highly of, and she had a great time in. And it was California still, in the Bay Area, which is really diverse. And so I was like, you know what? Yeah, I think I could do this. I could be a scientist. And I think that was the moment where I thought that I could actually do science that literally comes from my mind.
It's only after then where you actually started developing the type of scientist that you would become, right? Because that's where you join Caroline Bertozzi's lab. That's where you met proper chemical biology, and you learn about the bioorthogonal chemistry and all these things that basically characterize all your studies. Am I right?
Correct. I mean, Caroline Bertozzi is such a force in the field of chemistry generally, and certainly chemical biology. I knew of Caroline before I met her. I was mesmerized by her approach. She was both a chemist and a biologist. She had somehow found a way to make chemistry be useful for biology. And as my young eyes saw it back then, and the applications were endless. And it's still true today. And I remember thinking, wow, she must have geniuses in her lab. Like all these really smart people working on really important problems. What I wouldn't give for 15 minutes with her. And so this was before I got into Berkeley. And then I got into Berkeley. And of course, my number one person I wanted to talk to was Caroline Bertozzi. And getting on her calendar it's incredibly tough, or so I heard, because when I emailed, she immediately was like, yes, let's meet, let's talk more about TB and all the stuff we could do. And we had a fantastic conversation about what had been done in the world of at the intersection of chemical biology and tuberculosis research, and what was left to be discovered. And I remember I told her, it's like, I have a mind to do something that could be directly applicable to the places that I left. And so these are the kinds of projects that I'm interested in. And she's like, well, that's what we'd like to hear. And so that's how I joined her lab. Mind you, I was still like a fangirl for everybody in that group because of the work that they were producing. It was so amazing to watch. I felt like I just magically stumbled into this thing. That Carolyn at any point is going to be like, wait, how come you're here? I really didn't feel like I deserved to be there. But Carolyn has this magical touch of bringing the best out of people. And I felt that. I really did. I felt encouraged. I felt listened to. And she was really interested in the ideas that I was proposing, which sometimes would surprise me because even I didn't believe in my own ideas. But she did. And she would actually listen and encourage and give me leeway to explore what it is that I wanted to explore.
That's what a great mentor does, right? That's what a great manager does.
Yeah. And I mean, I ended up taking my project in a direction that was vastly different than what she typically does in her lab, right? Like some of my work ended up becoming clinical. Like we went to South Africa and we did a little clinical trial, and we are doing more clinical trials because of the work that I did, and she had never done that before in her lab. And again, it's the beauty of having a great mentor is the fact that she allows her students to explore their interests and see what you can do out of that.
And it's time to jump into the science, right? Because now you're excited, maybe giving a little bit of hint into the importance of it. It's fair to let you describe what your project has been so far and what you focused on.
Yes, absolutely. So Carolyn's lab is based on this concept of bioorthogonal chemistry. And the idea there is that you have, you can engineer chemistry that can occur in a biological system without disturbing the system. So imagine you want to visualize a particular biomolecule in a cell. You can add onto that biomolecule a tag that will selectively interact with a reporter that you feed into the solution. And they will interact together in a way that does not perturb the biological surroundings. And then you can precisely and selectively monitor the biomolecule. That definition, that basis has been a breeding ground for lots of different technologies in lots of different applications. In my case, I leveraged this biomolecule called trihaloose, which is a sugar that is metabolized in Mycobacterium tuberculosis, the pathogen that causes tuberculosis. Is metabolized and inserted into the cell surface. And so what I did, I engineered a trihaloose dye. So I essentially just put a fluorescent molecule onto trihaloose and then fed the cells that molecule, and that essentially made the cell surface of the mycobacteria glow green under fluorescent microscope.
We hope you're enjoying this episode of Bringing Chemistry to Life. It would really mean the world to us if you shared what you like with a couple of your friends or colleagues. A fun way to spread the word would be requesting your free Bringing Chemistry to Life t-shirt. And if you want to find out more about Mireille and her work, you can download additional information and content recommendations. Stay tuned at the end of the episode for information on how to do it. And now, back to our conversation. So, is this selective for the mycobacteria because mycobacteria are explicitly using trailose, metabolizing it, and then excreting it on the surface. Is that what makes it selective for that mycobacteria, or is it this is a dye that is gonna kind of bind to several different microorganisms?
Yes and no. So the trehalose is just two glucose, connected to each other. Trehalose molecule is used in a variety of different ways in a variety of contexts. Like plants use trehalose as a source of energy. In mycobacteria, they have a pathway for that, but they also, in addition to that, have a pathway where they use trehalose as a building block to build the cell wall. So what they do is they lipidate, so they put lipids onto the trehalose, the two glucose molecules, and then these lipidated trehalose then get inserted into the cell surface, and that's sort of like part of the complex architecture of the cell surface. That's unique to mycobacteria. So this is a class of organisms that include Mycobacterium tuberculosis, Mycobacterium leprae. So they cause a wide range of illnesses around the world, and they have this unique ability to lipidate trehalose and put them on the cell surface. It also is what makes them very challenging to tackle, is because this lipidated trehalose layer is very thick and waxy and hard to get through.
So you feed the microbes the building blocks they use to build up this outer wall. They synthesize this glycolipid out of it, and then put it in there, as use it as a brick in their external wall. The only difference here is that you actually modify the trehalose with a dye that then makes the cell fluorescent, and then you can visualize where the actual cell is. And I suppose the fact that this only happens if the cell is actually actively building the outside wall, so the cell needs to be alive. Is this what makes your method different than whatever other colorimetric methods that are out there?
That's right. The trick to the dye that we selected is that the dye itself doesn't fluoresce on its own in a aqueous environment, so like in water and stuff like that. It only becomes fluorescent when it's in a highly hydrophobic environment. And so this unique pathway of trehalose lipidation, when the trehalose dye conjugate, so we call it DMN tray, when DMN tray gets inserted into the cells, it gets lipidated by way of the traditional trehalose metabolic pathway and then inserted to the cell surface. And this whole time, during this whole process, DMN itself, the dye, is non-fluorescent. But as soon as it gets lipidated and inserted into the cell surface, that's when it turns fluorescent, turns on fluorescence. That's the key change with this dye, is the fact that because there is this turn on step to it, it makes the dye require active metabolism, right? In order for it to work, that's one. And two, it requires that the cells themselves are alive, that the cell wall has integrity and intact. So take it in a broader context, that means that the dye can tell you, one, is the cell actually actively building its cell wall? And two, is the cell wall structure intact? And which really means that, is the cell alive, and is the cell surface, like, is the cell there, first of all? Is the cell present, and is the cell viable? Those are the two things that it tells you at once. And so because of that key specificity to live cells, it makes this dye incredibly interesting, because then it can tell you if cells are dying in the context of drug treatment. So suppose you have a patient that has TB. They come in, they give you a sample, you use our dye to visualize how many TB cells they have, and then you give them a drug, and over the course of their treatment, you just monitor the number of cells that are viable in the sample. And hopefully what you want to see is that as people are getting treated, the number of cells that are viable goes down. That means that they're able to fully cure their illness, or at least get rid of the live cells. But if you don't see a change in bacterial load, as we call it, then maybe these dyes are resistant to this drug. And so you can actually potentially detect drug resistance much earlier than other methodologies can. And the only way we have right now to detect the drug resistance is to do culture of the TB samples that you get from patients. And culture takes weeks to get back. With this dye, you can quickly assess, like within hours or even within days, if someone has a particular strain of TB that is drug resistant. And you don't need to do one drug at a time. You can do a bunch of drugs at a time. It can be a multiplex assay.
What I like of this whole idea is its beautiful simplicity. It's super elegant.
Yeah. I will say that this isn't an idea that came out of thin air, right? There had a lot of work that had been done before my time. But no one had really thought about using this in a point of care specific field application. Because my eye was towards, I want a dye that can work with minimal processing. So I want a dye that anybody can use, that my grandmother can use, right? And because of how it functions, the dye itself that I have, because it stays dark up until it meets a TB cell, all you really need to do is put a drop of the reagent onto the sample. It's as simple as that, and then you wait. So I think, again, it's because of my perspective of trying to find something that could be directly applicable now, as quickly as possible in these environments. And because I had an understanding of what the resources are in those environments, I was able to try to take a technology that people had built for other scientists and make that applicable for point of care diagnostics of TB.
Just sticking on the chemistry, what is DMN, which is your dye? What does that stand for?
DMN stands for dimethyl naphthalamide. It's a naphthalamide-based dye that has this unique feature of being able to sensing the hydrophobicity in its environment, and therefore quenching or releasing high quantum fluorescence yield. It's not the only dye that can do that. There are a wide range of environment sensing dyes out there. I think that the key part here was using these classes of environment sensitive probes to be able to build this technology was the most important part.
And how stable and cheap DMN 3-allose is? Because I guess this is key, right?
Very key. DMN is this really small molecule. It's easy to make, and it's highly stable at room temperature, and of course in cold temperatures it's much better. Like, I ship my dyes to all my collaborators at room temperature, and it works just fine. It's a highly stable molecule. We really lucked out on that one.
Well, so you brought this to the next level, right? You mentioned you did clinical studies, so this is demonstrated to work. Is it actually currently used in field diagnostics?
No. So I got my PhD 2 years ago. And so we started the company, and about, let me say, close to 10 months after we started the company, as we were just gearing up to do the clinical trials, COVID-19 happened. And all the resources that were available for TB research have been rewired for COVID-19. I mean, anything you can think of that could be applied for TB has been used for COVID-19, because now we have a crisis. And for a very young, small company that is trying to do something better in the TB space, it was just the worst possible timing. Hopefully we can sort of get back on the train and do the clinical trial that we've always planned to do, and build the drug sensitivity assay that we've always planned to do. Now that the attention, hopefully the attention will get back to the other infectious disease areas.
I wish you best luck for it as well, because you deserve it, and the world deserves it in a way, because this could potentially change life for a lot of people. So we all want to see this happening.
Right, exactly. And I I do think that there's beauty in connecting with people in other spheres, right? Like connecting with the clinician that has, patients in his ICU, or connecting to the community health worker in India who's trying to find the patients and trying to diagnose COVID-19 quickly. I think all of those stakeholders are informing the work that we do. And it was vital for COVID-19. It was vital for vaccine development. And so at least in my view, I see the future of science being very much in close collaboration with stakeholders outside of scientists.
How do you leave the interaction with scientists coming from other disciplines, right? Or even non-scientists that are more or less involved in your project? Do you find it easy to interact with them?
I'm very easy to talk to non-scientists. And in fact, I make that a mission of myself so that I can stay connected to the community. I will say that academia wasn't built that way. It wasn't built to make communicating between scientists and the general public easy. In fact, our language is incredibly complicated. Like it's just, we make it incredibly hard for a non-scientist to know what it is that we do. There's so many movies out there where there's an evil scientist or a mad scientist. And I think it's just a reflection of the fact that the general public doesn't really know what we're doing. And we don't really do a good job at communicating that. And so I try to maintain an open door policy with people who are not chemists or biochemists, to listen to them, quite frankly, to just listen to what's going on out there and what it is that I can do on my end. And to also try to facilitate communications with other scientists and try to bring that to light, particularly when it comes to social justice as well. This is an important part. Opening the door to understanding how we behave is just similar to how the rest of the population behaves, makes communicating with everybody much easier.
So what's the future for your technology and Olilux Biosciences and your diagnostic kit? How far are we from really seeing this applied in the places where it is needed the most?
We are working very hard to make this technology available in the field as soon as possible. What's on the horizon? We are gearing up for clinical trials and building this drug sensitivity assay. We are gearing up to set up collaborations internationally across the globe. I hope that more people care about TB now, and I hope that listeners or people who are interested in engaging with us do so. The future is bright in my view for the company. I think now that people have an acute sensitivity around infectious disease problems, I hope that the care we have for COVID-19 will also transfer to other infectious diseases and understand how a major problem they've been in the past and continue to be. And I'm excited to embark on this journey with, I wasn't trained as a business person, as an entrepreneur, but here I am. And I'm excited to embark on this new direction of my career, understanding how difficult, what the challenges come with trying to take a technology out of academia and make it applicable in society.
And I'm sure there'll be a lot of solution and smart ideas coming from you. And I'm actually gonna keep an eye on what you're gonna do in the next few years, and hopefully interview you again. So as we come to the end of our chat, which has been brilliant, by the way, I have one question that I always use to close my interviews. Now that you are at this point of your career, you're going through a lot, and there's still a lot to do. But if you had to stop and look back, what would be one piece of advice you would give to someone else just starting, right, in their career?
When I look at what my path so far, I think resilience is the word that comes to mind. There's a lot of struggles that I had, a lot of challenges, and a lot of winding roads. And the only key part, like you said at the very beginning, the only key part was the fact that I saw an opportunity and I was able to leverage that opportunity. And that requires resilience, right? Like if you're charting a new path, you have to be resilient and keep at it. Believe in the work that you're doing and be resilient at it, and in your own motivations and passions.
That was Dr. Mireille Kamariza, Junior Fellow at the Society of Fellows at Harvard University and one of the Chemical and Engineering News's Talented 12. Thanks for joining us for this Season 2 episode of Bringing Chemistry to Life, and keep an ear out for more. If you enjoyed this conversation, you're sure to enjoy Dr. Kamariza's book, video, podcast, and other content recommendations. Visit thermofisher.com/bctl to access these recommendations and register for a free Bringing Chemistry to Life t-shirt. You can find the URL in the episode notes as well for an easy one-click access. This episode was produced by Matt Ferris, Matthew Stock, and Emma Jean Weinstein.